My name is Seth Martin, and I am a professor and preventive cardiologist at Johns Hopkins Medicine. Well, I think it means they're going to have more accurate LDL for their clinical practice. So LDL is front and center in our clinical practice, and it helps guide decisions around medicines that reduce ASCVD, atherosclerotic cardiovascular disease events, and reduce death. So it's literally decisions that we're making that could be a matter of life or death for our patients. And so we want to have the most accurate results. And so I'm thrilled that by providing this new machine learning version of my equation, that we can scale up accurate results even more widely. So that this machine learning version is not something that's going to be a dynamic model that's changing over time. To be clear, it's a very transparent, fixed equation that'll get coded into lab. So it'll year after year, you'll continue to get the most accurate results. I think with the original equation and now with this equation, we've basically optimized LDL cholesterol calculation. So what was really cool is that the new machine learning version basically gives results that are similar in accuracy to the original equation that we had published over a decade ago, which was developed in more of a manual fashion. So I think that for pharmacists practicing, they can just will be able to rely on more accurate LDL results, and pharmacists and other clinicians are very busy. And so I think it's quite important here that we get results that are automated in the lab. So the whole point of this new equation is to make it easier for labs to automate the calculation so that when they get to you, they're trustworthy results. Yeah, it's a great question. The key clinical issue is that the original Friedwald equation, as well as other equations that have been developed, tend to have an issue of underestimating LDL cholesterol levels. So they may give a result that's falsely reassuring because it seems like the patient's LDL is really this low, but in reality, it's 10, 20 milligrams per deciliter higher, and they're actually above a clinical threshold where they would qualify for evidence-based lipid-lowering therapy, including statins and non-statin therapies such as ezetimibe, bempedoic acid, PCSK9 inhibitors. So this change to a more accurate equation corrects for that underestimation and now will allow those patients who to get a better representation of their LDL cholesterol levels and to qualify for these therapies that reduce risk of cardiovascular disease. So we're talking about saving heart attacks and strokes and saving lives. Yes, this is a great question. The stress test of LDL accuracy is in patients who have lower LDL levels and higher triglycerides. So if you see a lipid panel, for example, in someone you're treating with very high risk ASCVD and their LDL levels coming back on the lower side, let's say your goal is less than 55 and you get an LDL of around 50, and their triglycerides, let's say, are in the like 2 or 300 range, that's a type of situation where I'd be very wary of which LDL cholesterol equation was used. Because if it is the Friedwald equation or it's the Sampson NIH equation, that LDL level, I would be concerned is underestimated, and actually they're not at goal. And if you were to use the original version of my equation or the new machine learning equation, you would find that they're actually above that 55 goal, and they would still qualify for additional therapies to reduce risk of heart attacks and strokes and to reduce mortality. I think it's important to keep in mind kind of what we just said about knowing which equation was used to calculate the LDL results. And so you may, if you're getting a lot of results from your local laboratory, hopefully it's clear what equation's being used. Otherwise, it's worth checking in on that. If you're getting results from Quest Diagnostics, it is using my equation. but other labs are using different equations. So I think being cognizant of that is really important, and serving as kind of a local champion for change if it is needed to make the switch. And then I think being really cognizant of that issue as it pertains to LDL goal achievement, being we in our 2026 dyslipidemia guideline, we have very clear LDL goals of less than 100, less than 70, less than 55 based on the risk status. So knowing where one is, and then making sure that you have the most accurate LDL results in relation to those goals. And then being mindful that in our guideline, we do go beyond LDL to also have non-HDL and ApoB goals that can be used to further confirm that therapy has been optimized. I'd like to just thank you for covering this because this is such a key issue. Cardiovascular disease still is the leading killer in the US and around the world. And I think what I would like to do is just acknowledge the very important role that pharmacists play. My wife is a pharmacist. I recognize the very important role that pharmacists play in clinical care, and we're all in this together on the front lines of clinical care, addressing the leading killer of people. And we're at a time where we have very effective therapies, and if we implement these well and we use accurate LDL equations, I think we can continue to build momentum in reducing the burden of mortality and morbidity due to cardiovascular disease. So I'm very hopeful for the future, and I just want to recognize, I think, the very key role that pharmacists play in delivering this evidence-based care and saving a lot of lives.